Hey guys, and welcome back. I'm assuming that if you are watching this video, you guys either don't have the ability to access a gizmo via your phone or computer, or you are trying to figure out how to get onto the Explore Learning gizmo through the Clever portal and are having a difficult time managing that. So I'm going to go ahead and help you out either way. If you just want the Clever portal part of the video, this is going to be the first thing I do. If you want the whole thing, you can go for it. I'm of course not going to provide the answers, but I will walk through the simulation so that you guys can see it being walked through and done, and you guys can go ahead and finish some of the stuff that's required. So the first thing you want to do is when you go to the assignment on Google Classroom, it'll look something like this when it's done. We're almost done with it. And you're going to go ahead and access the Google Doc that is attached, Meiosis Simulation Exploration, and that's going to lead you to this right here. There's an introduction where I talk a little bit about what we've learned so far, so just a brief summary of what was talked about in the lecture. And then in the instructions it says we'll be going through a simulation of meiosis to better understand how it works. So let's go ahead and go to that simulation. So to access it, it says we need to use the Clever portal. If you guys don't remember the Clever portal, I'm going to go ahead and help you out with that right now. So I'm going to go back over here. I'm going to save this as a draft. You guys will see this as the actual assignment once it's completed. And we're going to go down to the syllabus in general class info. And oh my gosh, it's not here. Well then, I'll go ahead and make sure that I move that to here. Wow, I can't believe I didn't do that. Well, anyway, so how do you get to your Clever portal? Well, here is how you do it. You go onto the great Google, and you type in the work web page, the work website, work.nn.k12.va.us. And once you're in here, you're going to go to your Students tab. And under Students tab, you're going to scroll down to Clever. And you're going to click on Clever. And Clever is going to come up with login with Google or login with Clever. Login with Google. Okay, and once you're in with Google, it'll ask you to sign in using your NNPS Google account. I'm going to sign in using mine. And ours might look slightly different because mine will be the teacher. Yours, you guys will assumedly be the student. But once you get in here, it should look something like this. And what you're going to do is you're going to want to scroll down until you see Gizmo's Pilot. It should be down there somewhere on that page. You're going to click into the Gizmo's Pilot, and that's going to lead you into your actual page. Now, yours will not look like this. Because this is my teacher portal, but yours would look like this, so it'll lead you straight into AP Bio 5. You'll have to probably click on my name and indicate that that's the class that you're in. But once you're in here, you're going to go down to the Meiosis gizmo and click on that, and then click on the gizmo to go ahead and launch it. And when you launch the gizmo, you are going to be answering the questions that are down here. Some of these are the pre-knowledge questions, so you'll answer those before you start. And then you're going to start following these instructions step by step. So the first thing we're going to do is run through meiosis, and we're going to run through it in males. Because remember, males and females, this is the process of making their sex cells. It's going to be a little different. And guys versus girls. Guys, we're making sperm. Girls are making egg. So we're going to start off with the guys. And we're going to go over here to male and click on male. Okay, and so that's going to go ahead and sort of change everything that's going to happen. Now, if you notice over here, we've begun interphase. Okay, so we've gotten our interphase. We're growing. We've increased our DNA information, right? We've copied all our chromosomes during the S phase. Whoops. Here we go, chromosomes are copied during the S phase, and then we have the G2 phase where the cell continues to prepare for division, it synthesizes new proteins, and gets bigger, right? So all that stuff we learned in interphase, it's 90% of the cell's life. Okay, so in order to make it grow, we're going to click on the DNA, and there's interphase done. Woo! Okay, that was fast. Okay, so now during prophase 1, we're going to go ahead and break down the nucleus because that can't be in the way when we're dividing our chromosomes. Remember, the nucleus protects chromosomes and DNA, but we don't want it there now because we're moving out of our house, so we're kind of getting rid of that. Okay, and now we're going to go ahead and condense our DNA into chromosomes. So here's that coiling I told you guys about before. Okay, and now that they're condensed, we're going to go ahead and drag our centromeres into place. If you guys don't remember what a centromere is, it's just that little organelle that shoots out those protein fibers that attach onto your chromosomes and help to split them apart, right? So and these were centrioles. Sorry, I don't know if I said centromere or if it said centromere, I apologize. Okay, so anyway, we're going to go ahead and match our chromosomes up based on their homologous pairs. So here we have this chromosome looks like this one, so I'm going to match them up, and this one looks like this one. Those homologous pairs have been doubled. You can tell that, right, because there's two of each. So these are the sister chromatids in each pair, and we can see that the lighter pair maybe came from dad, and the darker pair came from mom in both cases. So maybe this is chromosome 1, and this is chromosome 2. Obviously, when we're looking at a real human cell, we would see 23 of these pairs, but here, for simplicity's sake, we're seeing 2. So we know that we need to go ahead and start dividing them, but something needs to happen first. We need to have crossing over occur, because remember, we want each of our gametes or sex cells at the end of this to have a different formation of genes. We want a different combination of alleles. So in order to do that, we're going to cross over. Now, normally what would happen is that these guys over here would come, and in real life, they would sort of twist around these guys, and they would swap information. And these guys would swap information in whatever. But in this simulation, you just click in. And you can see, so we swapped information. So the light green swapped with the dark green, and the light blue swapped with the dark blue. So cool. There we go. So we've shuffled our alleles around. We're good to go. We're going to go ahead and continue. All right, and here's the metaphase plate. So this is where we're going to line up the chromosomes. So here's the second place where we can start getting that genetic diversity. So crossing over was the first step in getting that genetic diversity, but here is going to be the law of independent assortment. I could put these guys over here and these guys over here, and the law of independent assortment states that it's random as to which one of these sets of homologous chromosomes will make it into each one of the cells. So for instance, in this case, I'm going to drag my little fibers, my protein fibers from my centrioles, or centrosomes as they're saying, out to the centromeres of the chromosomes, and I'm going to go ahead and split them apart. Okay, and the law of independent assortment states that it's random as to which one goes to which side. So here I got the mostly dark green with the light cap and the mostly light blue with the dark cap. And over here I got the mostly dark blue with the light and the mostly light green with the dark. It could be, this is not the way it happens every time the cell could divide. So that just happened by chance. It could have been that I got the dark blue one over here and the light blue one over here, or vice versa. So it's random as to which chromosomes make it into which of the new developing cells. And that's what the law of independent assortment means. And that's that second part of that genetic diversity that I talked about in the lecture. So here we have our sister chromatids are still together, but homologous chromosomes have now been separated. And we're going to go ahead and we're going to reform our nucleus and undergo, so that's telophase. So we start getting our nucleus back, and then we're going to do our cytokinesis to go ahead and split the cytoplasm of the cell. Now we have 2 new cells. So that was the end of telophase 1, and we are now entering into prophase 2 of the second round of division. Notice that we are not doubling our DNA before we just dive right in, and that's because we want a half count of DNA or a haploid count of DNA in our new cells. So this is prophase 2. We're going to condense our chromosomes just like before and get rid of our nucleus. Only this time you'll notice that we're not going to perform crossing over again. That's just happening just the once, okay? So we're going to then drag our centrosomes, as they're calling them here, or centrioles over here to either side. And we're going to go ahead and drag our chromosomes to the metaphase plate. And this time we're going to separate the sister chromatids from each other. So here I'm going to go ahead and attach my spindle fibers. Attaching spindle fibers to the centromeres of these chromosomes. And now we're going to go ahead and click and divide. And then we're going to go ahead and reform our nuclei. So that was anaphase. This is now telophase. We're reforming our nuclei. And now we're going to go ahead and do cytokinesis on both to split them. And we're going to reform our chromosomes. And here we have 4 genetically distinct sperm cells. Okay, so that should answer all of the questions for activity A, if I'm remembering correctly. So this is all activity A. We talk about it. Now, activity B is going to be, we're going to compare what we just did in the sperm to the egg. So we're going to see whether or not the egg differs any at all from this. So we're going to go ahead and press reset. And this time we're going to press female, and we're going to do the same thing. So I'm just going to kind of click through it. We're going to see we got the nucleus disappears. All right, so prophase. We're going to go ahead and condense our chromosomes down into, or DNA down into chromosomes and chromatin and chromosomes. We're going to move our centrosomes to their proper locations, and we're going to go ahead and line up our homologous chromosomes. Okay, they are going to go ahead and perform crossing over, and then we're going to go ahead and line them up at the metaphase plate. All right, and here we're going to have our centrosomes attach those little fibers onto the centromeres of the chromosomes, these little pieces in the middle. We're going to pull them apart using anaphase 1, and then we're going to go ahead and reform the nuclei to finish telophase 1 and cytokinesis. So far, it's pretty much the same, but I want you to notice that this one got more cytoplasm than this one. We can see it's a bigger cell than this one is, and that's going to continue. So we're going to go ahead and do prophase 2. No copying of the genetic information, just straight on through. We're going to go ahead and drag our centrosomes to the correct locations. We are going to go ahead and line up our chromosomes on the metaphase plate. We're going to attach our spindle fibers onto the centromeres of our chromosomes. And we're going to pull apart our sister chromatids. Okay, and then we're going to go ahead and reform our nuclei. And here we have our nuclei. We're going to go ahead and split down the middle. And notice that, again, the split was not equal. We actually have 3 small cells now called polar bodies and 1 ovum, which is the mature egg cell. So interestingly enough, in females, instead of producing 4 egg cells, each time that we have this process happen, we only produce 1. Why is that? So females actually start off with tons, thousands and thousands, about 40,000 actually, if I'm remembering correctly, precursors to egg cells in their bodies. And those are actually developed when females are in a fetal form, so before you're even born. That's very different than males who only develop their sex cells and those when they enter into puberty. Go figure. But so females, they have this stuff and from the fetal stage on. And the egg cells are basically whittled down and whittled down. So what's happening is to increase the genetic diversity, the egg cells have to go through this process of what we just saw, of the creation of polar bodies, which are destroyed. And only 1 egg cell sort of survives. It's kind of like the Hunger Game for egg cells, and this is to ensure that the egg cells that survive are healthy, they have everything that they need, and that they're genetically diverse and less likely to be copies of the ones that did not survive. So that's pretty cool, and it ends up, so we end up getting 1 mature egg cell, whereas in males we got 4 sperm cells. Okay, so that should be enough for you guys to be able to answer the questions. There is another cool thing about the size difference, though, in the egg cell. So if we were to compare the egg cell to the sperm cell, the egg cell is way bigger than the sperm cell. And this is because the mother's body is going to be the one to actually be nurturing and growing the child. So the egg cell has... most of it has this very nutrient-rich yolk inside of it, like an egg yolk. And that egg yolk is going to help to power the metabolic processes that are going to go ahead and start creating the child once the sperm fertilizes the egg. So hopefully that has helped you guys out with this simulation. Go ahead and answer any of the questions that are associated with the model. I hope you guys enjoy it. And if you have further questions, please do not hesitate to email me, and I'll do my best to help you. All right, guys, take care.